So now I'm going to talk about prokaryotic transcription termination, of which there's row independent and row dependent. That's just a protein. In row independent, row P is not used, but instead these things that are known as hairpin loops form because of inverted repeats. Now in row dependent, row protein is used, and so what will happen is that the row protein will bind to the nascent mRNA, and then it'll kind of move up the mRNA until it gets to the RNA polymerase, and that'll cause the RNA polymerase to detach and then become free in the cytosol. So you can see that we have our free mRNA, and then the row protein will also detach after this and be ready to be used in another reaction. So back here over at row dependent, you might be wondering what I meant by innervated repeats. And basically what I'm talking about is these little green sections that I have marked here. Each of these is an innervated repeat, so there's an innervated repeat. And what the repeats means is referring to these green sections. If you were to read out from the middle of each green section, you would see that the C's and the G's complement each other. And so when that is transcribed, you can get down here, and we can see their nascent mRNAs kind of like folding on themselves, and that's because of up here, you can see that the C's and the G's that are complementary, they've kind of bonded to each other, and they would do that throughout the whole innervated repeat, forming the hairpin loop. And the C's and G's will be holding up these little hydrogen bonds that go throughout the entire hairpin loop, and the formation of the hairpin loop signals the RNA polymerase to stop and terminate transcription. And it's worth noting that these C's and G's can be replaced by A's and U's, vice versa.